Hey guys, last video for chapter 9. This one's going to be on fermentation. So this enables cells to produce some ATP without the use of oxygen. There's times when your body runs out of oxygen. So if you think about if you're an athlete or really if you've been in gym class in general, there's a point in your exercise where you start to feel really tired. The reason why you feel tired is your muscles have run out of oxygen. Okay, so what your body has done is it's converted to a process called fermentation. So it allows us to get some energy to continue working, but you can't work at your optimal productivity anymore. Okay, so cellular respiration relies on oxygen. It allows us to produce a lot of ATP, and when we can do cellular respiration, we can do quite a bit of activity. But in the absence of oxygen, I should say, cells still can produce some ATP through a process called fermentation. So if you remember, why can they produce a lot of ATP? What's oxygen's role? Well, if you remember back to the last video, oxygen's role is to accept electrons at the end of the ETC. So if oxygen isn't there to do that, the ETC shuts down and it's not using those NADHs, those FADH2s that were mostly created in the Krebs cycle. Okay, there was 2 that was created in glycolysis, yes, but most of them were created in the Krebs cycle. So because of that, when there's no oxygen present, the ETC shuts down and the Krebs cycle or citric acid cycle shuts down as well. So the reason why is there's no oxygen to pull those electrons through the ETC. So what we end up doing is only glycolysis. And then we do a process called fermentation. But as far as ATP production, the only thing that's making ATP is glycolysis. So if we review that, glycolysis can produce ATP with or without oxygen. In aerobic conditions, that would end up going to cellular respiration. But in anaerobic conditions, we're going to do a process called fermentation to allow glycolysis to continue. So what it does is it couples with fermentation to produce very little ATP. Now, if you think about it, what did glycolysis create? It was only 2 ATP in comparison to the 38 if we get to go through cellular respiration. But so why, you might think, why would we do this? Well, if you think about it, 2 is better than none, right? If you have no energy, your cells die. We have to have that constant influx of energy. So this allows our cells to stay alive when they don't have oxygen. It allows your body to stay functioning until it can replenish that oxygen supply. So fermentation consists of glycolysis, and then it must also have reactions that will regenerate NAD+, which can be reused by glycolysis. So what's the overall purpose of this? It's to allow glycolysis to continue. Let me repeat that. Fermentation's job is to allow glycolysis to continue. And I'll explain that with a diagram in a little bit. There's 2 types of fermentation. First type is called alcohol fermentation. This would be done in yeast, and plants do this as well. And what happens is pyruvate, which is a product of glycolysis, is converted to ethanol alcohol in 2 steps, one of which is going to release CO2. So if I were to diagram this, again, we have glycolysis. It's going to break glucose into pyruvate, but glucose plus alcohol fermentation what goes in? Glucose. That gets broken down into pyruvate, which then becomes alcohol plus CO2. Okay, those are the 2 products, alcohol and CO2. We also make, through glycolysis, 2 ATP. Okay, so those are overall, we make 2 ATP, alcohol, and carbon dioxide. Now, where did these come from? Glycolysis made the 2 ATP, alcohol fermentation made the alcohol and the CO2. All right, so that's one type of fermentation. Animals do not do this type, but alcohol fermentation is done by things like yeast and plants. All right, the other type is called lactic acid fermentation. This is what we do as animals. As humans, we have to do lactic acid fermentation. So pyruvate is reduced directly by NADH to form lactate as a waste product. So if I were to diagram that, again, what we start with is glucose. Still breaks down in glycolysis to pyruvate. Oops. Forgot a B in there. Pyruvate. And then that creates lactate. Okay, or lactic acid. So this process here is glycolysis still. And here would be your fermentation, your lactic acid fermentation. Now, we also again create that 2 ATP that is made in glycolysis, not in fermentation. So we make 2 ATP and lactic acid or lactate. So those are the two types of fermentation. You have lactic acid fermentation or alcohol fermentation. Both of them do essentially the same thing. Their products are just a little different, but their job is to allow glycolysis to continue. So how does it do that? This diagram is good at explaining. So here at the top, we have alcohol fermentation. Glycolysis happens. Here's our 2 ATP that is created, and we make 2 pyruvate. But remember, what we need to be able to do is allow glycolysis to continue. In order for glycolysis to continue, you have to use the 2 NADH that it made. So it has to do this. So what allows that to happen is the fermentation. So what does fermentation essentially do? It uses the 2 NADH so that it can have this 2 NAD plus so that glycolysis can continue. So the 2 NADH along with the pyruvate are converted into 2 ethanols along with carbon dioxide. Okay, so if we look at lactic acid, remember this is what we do as humans. Again, glycolysis happens, we make 2 ATP and pyruvate. We're going to use that NADH to create lactate in this case. Again, what's this job? It's just to allow glycolysis to continue because we at least get some ATP, which your body needs, your cells need in order to continue functioning. Both fermentation and cellular respiration are going to use glycolysis. Glycolysis continues to happen. So you're still going to be oxidizing glucose into making pyruvate, and you're going to get 2 ATP out of that. It's our only option when oxygen is not present. They differ in their final electron acceptor. So if you remember in cellular respiration, the final electron acceptor is oxygen. Okay. But in fermentation, the final electron acceptor is either that alcohol or the lactate. So it's just the final electron acceptor is the difference. So without oxygen, that energy remains in the pyruvate and really cannot be used because it can't enter the Krebs cycle. So pyruvate is the key juncture. It's like the fork in the road. Glycolysis happens. It makes the pyruvate. If oxygen is present, it goes through the link reaction into the Krebs cycle and goes through cellular respiration. If oxygen is not present, it goes into fermentation, and depending on the organism you are, you're going to either make ethanol or lactate through fermentation. So glycolysis occurs in nearly all organisms, probably evolved in ancient prokaryotes even before there was oxygen in the atmosphere.